We have our next speaker, Lennart Hilbert from the KIT in Karlsruhe, talking about Pol II clusters, etc. Welcome, Lennart.
Yeah, thank you. So I want to help everyone to warm up a little bit. So we're going to transcribe a gene together. You can see it here. The first thing we do is to get Pol II onto the promoter. When it's there, the first thing before it gets active is it takes a break. So we have pause Pol II. That's the first step. From pausing, you have to release it. So pause release takes you into actual elongation. So you can see here Pol II running through the gene, actually elongating RNA transcripts. And I think so far you're probably following. Important to remember, both steps are associated with a specific phosphorylation step of the CTD of the Pol II subunit 1. Where things maybe are little less familiar is if you consider that sometimes even about 90% of the pause Pol II get released before they ever go into the gene. So you have this still serine 5 phosphorylated Pol II floating around, and it even has an affinity for these, let's stay neutral, macromolecular clusters. And there has been a lot of work on coactivators and sort of liquid phase properties, which may be connected to this meeting, that has implicated some sort of liquid phase separation mechanisms in the formation of these clusters. I think rightfully there are many questions in the community about these clusters and liquid phase properties. First one being, how can you get a liquid phase condensate if inside the cell you actually have subsaturation concentration, so you don't actually cross the phase separation boundary? If you consider that these two phosphorylation states and states of transcriptional activity occur in different spaces inside of the nucleus, how does that go together? And maybe the one that I found most striking is when you look at these clusters, they don't look like droplets. They look very unshapely, unfolded, fuzzy, granular. So how does this go together? To address some of these points, I will bring in my favorite model system, the pluripotent zebrafish embryo. It provides you on the house about 4,000 pluripotent cells fertilized 4 hours ago, this organism. So it's pretty fresh. It's not cultured or anything. It's a vertebrate embryo. So I think there's a lot to like. You can also do microscopy in this very well. So here you see in live embryos with antigen fragments that can detect these two phosphorylation states, you can see very prominent clusters that occur inside here. So we get lucky, we get to investigate shape. And they occur in really various shapes, and we try to sort them a little bit. So we just gave them type numbers. So type 1 you can see is small and round and sometimes transcribed, sometimes not. So type 2 is a lot larger. And you can see in the second row that there is not so much transcription activity. Type 3 is like a bit crazy. So they're very unfolded in the recruited Pol II channel, and there is a bit more transcription activity present. So you see this in live embryo, so you should maybe believe this a little bit more than when it's fixed. But when it's fixed, you get more resolution if you put your STED microscope to it. Or if you go to Uli Nienhaus at Karlsruhe Institute of Technology, you get STED double depletion, which gives you even more signal and more resolution and so forth. So you can look at this in a little bit more detail. So we super resolved one channel and got in addition a second confocal microscopy channel that gives you the transcription level. So on the left, we get the structure quite nicely resolved. On the right, we also have the transcription level, so we can see the activity and correlate these things with each other. And we get the same types that we have seen before, but with better resolution. And for example, for the type 3, which is now looking like this cute donut, which kind of motivated that we even did the project, you can see, yes, there's a lot more transcription, but it's not actually inside of the area that contains the recruited Pol II, but instead it sits on the margin just outside. Keep that in mind. We, of course, recorded more than just one nucleus, but a good number, and we then put it to an automated image analysis, giving us here this map of geometry of all these clusters that we pulled out of the data set. And we have here area. I think area is pretty intuitive. Higher area means a bigger cluster. High solidity means that it's very round. Low solidity means that it's rather unfolded and has indentations. So you can basically put the different types on these gates. We put the gates quite extreme in the ends of the distribution so that you can pick out clusters based on shape. You can then transition the same cluster points into a map based on phosphorylation intensities as detected by immunofluorescence. And you can see that systematically the gated points actually show up in different locations on this phosphorylation map. So you can select by shape and get a different location in terms of the transcriptional control state. So there's a systematic correlation between shape and control state of these clusters. If this analysis seems a little bit fast, I agree. For the talk, it has to be fast. But the takeaway is you can see for high levels of recruitment, large clusters, type 2 and 3. And if you have transcription occurring on the margins of the cluster, you actually get them in these unfolded shapes. That's the type 3 that is unfolded. So I was alluding to liquid phase properties. Of course, we wanted to follow up a little bit on this as we saw all the papers on the topic. And wanted to look for hallmark features of phase-separated droplets, like, for example, coarsening. So here in this 20-minute time lapse that shows you the recruited Pol II in this nucleus, if for the 20 minutes you say nothing is coarsening, the pattern is not changing and everything looks stable, I agree with you. It doesn't agree with the plain liquid phase separation picture. If you look more closely into some more isolated cases of these clusters, you can see even stranger behavior. We call this split fusion cycles. So you can see how here first we have just one cluster, it splits into two, they fuse again, they split again, and they fuse again. I find it very interesting, quite striking. But again, you wouldn't expect this if it was just liquid-liquid phase separation. So there must be something else going on. We're inside of the nucleus. It's an enzyme that works on DNA. So probably we should be looking at features of the genome that might be involved in this. So this is based on already published ChIP-seq data. So we started looking here on the top track for polymerase 2 serine 5-phos distribution along the genome. And indeed, we found areas or parts of the sequence where we had very elevated levels of this mark. And they actually superimposed most of the time with areas or regions that contain high levels of histone 3 lysine 27 acetylation. You might know this is an active enhancer mark. And sure enough, if we looked into annotated enhancer databases, we found that this strongly superimposes with annotated super enhancers at this stage of development. You see on top, it says oligopane probes. Those are our DNA fish probes we ordered and produced to label these various regions. So we can also resolve them by microscopy. So here's the channel of the oligopane labeling for two of these super enhancers. In this case, they strongly superimpose and kind of dissolve in these Pol II clusters. And this happens when you do not have a lot of transcription activity associated with these clusters at the same time. Everything superimposed here, so they co-associate. This is what you would expect, looks good. You can have some of them sitting next to genes that occasionally activate and get transcribed. So you can see on the right-hand side, there's transcription activity. Then they actually sit right outside the cluster, as we expected. And when they're inactive, they actually go back into the cluster and kind of get immersed in it. Now, we've seen a couple of interaction rules. Typically, this is the point where I say we've seen enough to build a model. So we have this here. The model makes the assumption that we have these blue regions embedded in longer chromatin chains or polymer chains. So it's kind of a block copolymer model. And these can be visited by a red particle that can potentially form a liquid phase. You can simulate this on a lattice, as you can see here. We start with low affinity of the red particles. So even in presence of this kind of chain with the blue region, you only get little tiny bit of accumulation or adsorption. If you increase the affinity, you still don't get phase separation by itself, but you get clusters that form on these chains. If you increase in your model, hypothetically, the parameter of affinity even higher, you get this phase separation picture that you're used to. You get a droplet even without the presence of the surface, but you get the droplet just anywhere. And you also see it grows to maximal size. That's probably not what we want. So we choose here the case of so-called surface condensation. We didn't come up with that name. We only had the simulation, but luckily our colleagues pre-printed very early, so we knew what to call it. And we really knew how to understand our model. This is connected to in vitro work, much more detailed than what we have here, but I think we have the in vivo equivalent in the vertebrate embryo. So yes, so what is still missing in the simulations is to actually add these transcribed parts. So you can see here in these lattice simulations, there's gray parts to the polymer chains that are sticking out. And you can put more or less of these gray parts. You can put more or less of these blue parts. So you can basically create the ingredients of the type 1, type 2, type 3. Let your lattice model run and create synthetic microscopy images out of them. And you can really read them the same way in terms of recruited Pol II and elongating Pol II, and compare it to what we have had before. You can just visually see if you have more transcription, for example, you get this more unfolded shape as we see in the type 3. For now, you have to take my word for it. If you want to see the analysis, you have to look into the printed version. All right, so the last things I want to show is that we can also get back these sort of funny droplet behavior, non-droplet phenomena. So the first one is domains that do not coarsen with time. Yes, we get this from the simulation. So we get these clusters and then they do not coarsen much. Also, we get these here split fusion cycles. So the model also recreates this. It recreates basically everything else that I've shown and more, but I will not show all this. Instead, I will show the summary slide as I'm nearing the 10-minute mark. So you can see here, instead of the acknowledgement of the authors, I actually just put the author list. The work came out late last year, molecular systems biology, so pretty close to publishing office. The main takeaways, this structure here captures everything I've described. It has two rules that make it. The first one is that clusters form by condensation of Pol II and its friends on regulatory chromatin. This is how you get them. The second one is the clusters unfold because regions that contain a lot of elongation activity are excluded from the clusters. There's no way to satisfy this unless you unfold and sort of make these clusters a little bit unshapely. A picture from before Corona hit. This was happy times. This feels like a little bit of a return to this. I hope in the lab soon it's going to look like this again. Not quite, because we moved to a different lab on the same campus. But this is a lot of the people involved. You can see Tim, second from the right. Tim is also here at the conference. We arranged that he's going to join me at the table where he can talk to me and Tim after. Vasily on the right, Uli Nienhaus is not on here. Aga, the other first author, is also in the picture. Shut up.
Thank you so much, Lennart. Open for questions. Do you have an online question? So maybe I ask you, so how do you think in the...
Save me. Save me.
Oh, yeah. So in the, so what are the friends?
Yes, the friends, co-activators in general, we call this. So it, of course, mediator you would suspect in there because it makes contacts in many cell types or organisms. Things like chromatin remodelers, activating enzymes. Of course, the enzymes that deposit this lysine 27 acetylation. So that general class. And yeah, in general, I would say go back to the, I mean, don't put it back up, but in principle, go back to the references I've shown. There's so much work on what all is involved. Yeah.
And do you think that the local concentrations of those friends also make a difference for your different clusters that you're observing?
I think they will make a difference in terms of how big, or if you even get the clusters. That's one difference. And once you have them concentrated in the cluster and the enzymes are active and they work epigenetically to control transcription, how are they not going to make a difference?
Okay. Yeah.
Yeah, I really like this. Those movies of the sort of splitting and then fusing and events and stuff. I'm wondering if you think RNA could be playing a role in that as the concentration increases and then maybe decreases as you get further along away from this regulatory DNA. Yeah,
I think basically the results we have and the results that are known about that if you ramp up production of RNA, the production rate of RNA in these clusters and they unfold and then disperse is probably the same thing. Yeah.
I really like the pictures of the fusing and splitting, but I'm wondering from the simulations, it looks like they didn't really fuse all the way. And so like if you, with the, high resolution that you have in the simulated picture, you could see that there was really just like a small gap. But by like the simulated microscopy, it looks as if they were splitting. So I'm wondering if we need like different language to talk about things that are happening at the sub-micron scale.
Yeah, it's an interesting point. This shows up technically, but also conceptually for us. So whenever we segment objects, we have to decide on a threshold. And depending on where we put the threshold, we have objects that consist of many little parts, but they're obviously belonging together, or we put it down and it's one object. But also conceptually, you can ask when is something connected or not, and how does this connect to imaging resolution and so forth. Yeah, there's a lot to say, but no time.
I have two questions, one from me and one I can read off.
Only one.
You can.
Sorry.
The good one is from me. The other one's from no. You once made a statement where it said, "This is not condensation. This is the cluster." And we had this discussion over the meeting a few times. So, what made you be that strong about that statement, saying this is a cluster, it's not condensation, or vice versa? Where do you draw the line? You said that. We can taped, we can unwind, rewind it.
Yeah, I'm already swallowing the word that starts on S. But no, I think the distinction you can actually draw it quite sharply, and it's the CSAT, the saturation concentration. If you're below this, you may be
on a surface, or you mean the one?
No, wait. So the surface comes in a second. So one thing, so below this, you maybe get like very transient clusters, like Jay was showing, for example. But if you bring in the surface, those clusters can live on that surface for longer and also grow a little bit bigger. But if you don't have the surface, you're not really gonna get anything of size.
But the surface will always lower CSAT if you have,
That's another. I think that's terminology. I think you can say it like this.
But then you would say that there's never ever something like surface condensation because it's always below CSAT, because most surface condensations are below CSAT.
Yeah, I think you get condensation. you get something like a condensate below the saturation concentration if you provide a surface. If you do not provide that surface, you will not have that condensate at the same bulk concentration.
Okay, so then let me read out that question. We can follow up on this topic in the coffee break. It's actually interesting.
We will.
Is your model compatible with Pol II shuttling between transcription initiation condensates and splicing condensates by serine phosphorylation? Yeah,
I think if we would have imaged the splicing condensates or speckles or however you want to call them, then probably the elongating Pol II would be in this. I think we just didn't label it, and so we didn't see it.